Hi everyone, we're Team Diatom Biolab. Over the past 10 days, we built virtual cell foundation models for scalable discovery of synthetic lethality. We all know cancer is still one of the biggest global health challenges. Target drugs and immunotherapies exist, but responses vary widely because every tumor is genetically different. Choosing which gene to target and which therapy will work is still extremely difficult. But we do have one precision therapy success story. BRCA1 and 2 and PARP inhibitors. They work because of SL synthetic lethality. BRCA mutant cells already have weak DNA repel. When PARP is blocked, they die, while healthy cells survive. This is one of the cleanest and most powerful therapeutic strategies in oncology. So why don't we have more? The answer is scale. With 20k genes, there are over 200 million possible gene pairs, and we've validated only about 30k, a tiny fraction. Most experiments are slow, expensive, or impossible. And SL is highly context-specific, which increases the experimental burden. But a major shift is happening. Models like STATE, Tahoe X1, and LPM are trained on millions of perturbation response single-cell profiles. They learn how cells react when a gene is turned on, off, or edited, effectively acting as in silico experiments. This opens a new opportunity. Can these models help us discover synthetic lethal pairs that biology alone cannot reach? This question is not from nowhere. During my PhD, I did SL screening. The biggest bottleneck was obvious. Most gene pairs could never be tested. So at Diatom Biolab, our vision is to close the loop where AI proposes SL hypotheses and our automated bioserver validates them at scale. For this hackathon, we brought together a diverse international team from Australia, the US, and China to explore this idea. So we started with one guiding question. Do perturbation-trained virtual cell models can improve cold start SL prediction? To test this, we built a two-path strategy. We firstly adapted LPM and Tahoe X1 into simple do-input models that directly predict whether two genes form an SL pair. These serve as exploratory baselines for future true do perturbation modeling. And then we extract several types of gene embeddings from raw ESM2, STATE, Tahoe, and LPM perturbation embeddings. This capture either gene identity or how cells respond to perturbing that gene. We then compose the gene pairs features and use a strict AutoML pipeline to predict SL in a cold start setting, where neither gene in the test set appears during training. Together, these two passes reveal the strengths and limits of current perturbation models. In this setting, our perturbation and will embeddings clearly win. The best model, Tahoe perturbation embedding plus AutoML, reaches F1 about 0.68. When we switch to the non-perturbation Tahoe embedding, keeping everything else the same, the F1 score drops to 0.59. A name-owning state embedding is surprisingly strong. The score F1 is about 0.66, showing that some virtual cell models already encode used for biology in their gene token space. In contrast, generic baselines like protein language models, biomedical test embeddings, OPPI-owning graphs mostly stay in the 0.33 to 0.51 F1 score range, and a generic single-cell RNA model almost collapses in the cold start test. And our exploratory end-to-end dual-input models, adapted from LPM, Tahoe, do produce reasonable ROC curves, but their F1 scores stay near 0.41 to 0.51, clearly behind the other paths. The takeaway is that modeling cellular responses to single perturbation already gives us a strong signal, and the true end-to-end SL models will need that signal plus context and better perturbation encoding. So from this hackathon, we learn a lot that perturbation and will embedding do work, even single gene perturbation. Gene identity embeddings hit a ceiling, and no single model capture SL actually. And the end-to-end SL prediction, as I mentioned before, is possible, but require richer context. And looking ahead, the most exciting step is closing the loop. With Bioserver, we can test AI-generated SL pairs directly in cells, collect variability or transcriptomic data, and feed them back into our models. This creates a self-improving engine. And our long-term vision is simple. Virtual cells screen millions of pairs in silico. Bioserver validates hits in days, not years. And precision cancer therapies become routine, not real. So at Diatom Biolab, we are building the infrastructure for future biology.